Okay, I think we can start our seminar. We work here in Zoom and stream everything in YouTube. So I will switch it in that way. Okay, shortly, so first of all, thank you for attending this seminar in online form or later on in YouTube video. This seminar shortly about this series of seminars at we had conference on different topics, plant physiology, consciousness, physical chemistry, I mean a very broad spectrum. And then we decided to make a series of seminars, online seminars. Hope to extend it to more larger workshop as well. And this is one of the first series in our seminars, and we devote the whole series to plants. Different aspects of plants, sensing plants, plant physiology, I mean different aspects. And I will open the first seminar. We have already 2 or 3 upcoming seminars on the same topic on plant and phytosensing, plant stimulation, so different nice approaches here. And today I will speak about some dedicated points. As first of all, of course, we work with phytosensing. I will introduce a little bit in the phytosensing, different areas of phytosensing, but also a new topic is of course quantum. And quantum now it's not a sort of science fiction. We see, I will demonstrate quantum technologies enter in the agriculture. Largely in agriculture, there's something new, and I will try at least to give some overview over these different approaches here. Okay, shortly, also to mention, we published and are publishing different tutorials, different videos related to phytosensing in general. Just a moment. Okay, and here, for instance, some topics related to introduction to phytosensing, how to, for instance, to install electrodes we just published. Just a point here is so we will try to update you, of course, with all these latest developments, latest presentation, latest topics. Keep tuned in our channel. Okay, to my presentation, I will try to go in 4 areas. First of all, speak about 4 different application cases. I think they describe more or less why do we need plants, why do we need sensing in plants. The second important point, I will stress this many times, whatever technology you connect to plants deliver only some data, numbers, they have no meaning. Here it's very important to have biological models. So if we have biological models, then we can work with phytosensing, and I will try to introduce, let's say, that models that we use. And of course, how to implement with phytosensor and how to implement with different, let's say, topics around phytosensing. Some short overview about separate sensors, and of course, the last point, but it take almost half of the presentation about quantum effects, and is one of the most important things here, how to measure them. So it's not only a sort of science fiction, so future and nice. I will show we can really touch them and use heavily in our applications. Okay, first point, why do we need phytosensing and what are application cases of phytosensing? One of the largest, let's say, case of phytosensing is plant if we measuring different physiological processes in plants. So, for instance, precise agriculture, So there are different chemical or non-chemical approaches for stimulate plant growth, and we have to characterize this means. So how much it stimulate, what are boundary condition, which system of plants is affected. So we need to measure this the physiological processes in plant. Same story, plants have stress. There are different pests in plants. So here's the same story. If we see some, let's say, diseases in plants, typically plants are going to die at that time. So we need to know this in advance, so some days or even weeks in advance, to take some means to prevent this. So there's only one way. If we're measuring physiology, we're measuring stress, plant stress, and we can see, okay, something is happening or is going to happen, or plant is going to die, and we can prevent it. This second, for instance, case. Other case, we use plants as biosensor. I will speak about this later. We also, in this case, we have to measure different approaches, different processes here, and so we also have need access to plant physiology. This is, let's say, one of requirements of this application cases. We, at least in our system and in state-of-the-art approaches, we have access to all main plant areas or organs or subsystems like leaves, canopy, biomass, stem, soils, root. Some advanced sensor like nuclear magnetic spectroscopy. We'll also speak about this. Generally, in the community, we differentiate cases, soil-based production and soil-free production, so with different sensors. But here is one of the largest approaches we measuring, because we need to know how is going plants. We need to know well-being status of plants, some plants' health, and of course to do something with this. Important is here we are able not only to measure but to control. Typical phytosensing system can connect this power management modules, and so we entering in the second application case. This is related to biofeedback sensing. Why it's needed? This is mostly in B2B cases, so business-to-business collaboration in such greenhouses, some vertical farms with production. Typically, they use periodical protocols, so you can put different times here, and they periodically switch on, switch off light. Point is, it's work well, but it's not optimal. It's not optimized to specific plants, to specific condition, to stress of this plant. And so, idea is here we take data from plant physiology, soils, environments, we process, for instance, with artificial intelligence here, and put it back to different actuators, and we can optimize growth conditions. Just to mention, optimization of growth give huge effects. For instance, wheat, in normal condition, wheat productivity is about 4 tons per hectare, but in vertical farm, it make about 700 tons per hectare. So it's over 200 times more. So all this investment, also return of investment is very fast here. Optimization of productivity, removal of stress in such system. There is one of, let's say, cases what we encounter now. Next point, next case, and at least for indoor application, there is one of the largest application cases if we use plants as biosensors. Why we need plants as biosensors? First of all, sometimes plants are extremely sensitive. This is one of the main points. Plants are sensitive to weak environmental stimuli, weak magnetic fields. For instance, plants can sense magnetic fields in range of about 50 nanotesla. And this over normal variation of magnetic field. So there's huge sensitivity, and sometimes, for instance, ozone level, plant can sense something like 10 ppb. Not all technical sensors can measure this. But most important, plants are able to measure biological effect of different pollutants and contaminants. So we cannot measure this in technical way, only by using biological systems. And here with plants, they measure, for instance, a combined effect of ozone as pollutant and high electromagnetic emission. And so in this way, we can really sense what is happening if we have high ozone level and high electromagnetic stress, or some other pollutants and something else. So typically, for instance, we had a large project for measuring such combined biological pollutants in the city, in urban environment, and this is one of, let's say, application cases. Another point is here is AI phenotyping. So phenotyping is characterization of plant parameters under different conditions and different behavior. And for instance, here we can use this approach and the system for phenotyping. The system produces a large amount of data. It's very simple to use. You have just this, we call it a biochemical laboratory and new table, and this produces continuously data. You can train your neural networks. No, but common case. Here also to mention all these human-plant interactions. For instance, how human interact with plant is also fell in this application case. And lastly, but in currently did give more and more, let's say, importance grow of this application case. There is a called water problem. Point is that this climatic changes means less and less water. Period between, for instance, rain increase. In Germany, we had last weeks about 6 or 7 weeks without rain, then 2 weeks of rain, and then again 6-7 weeks without rain. So this long period without water for plants is difficult to survive, and so currently all these farmers, agriculture enterprises, they need to adapt plant to this period. So there are many different approaches here, how it can be used. Of course, improvement of irrigation system, but one dedicated approach is deficit irrigation. It means you just remove, you reduce amount of water for irrigation. This results in less yield. But generally, if you calculate yield over water, so it produces more. But here problem is plants can easily, hydrodynamic system of plant can die easily. And for instance, it's important to monitor hydrodynamic system and to prevent this degradation of hydrodynamics. So it's, for instance, one of cases what we use, and also improvement of this water versus yield ratio. It's also one of these application cases. So totally, if I conclude, there are 4, let's say, application cases which requires sensing of plants, different data, biological data of plants. And now the question is how to do this, how to, let's say, apply different sensors, how to install it on the plant. And I start with one of really important point. I stress it again and again. All these different, let's say, sensors, what you put on the plant, provide you only numerical, let's say, numbers, or they have no meaning. To understand this number, you need biological models. At least in the community, and of course in our system, we use 4 biological models. We label them with this red, blue, green, and orange, so we can use it directly with measurements, or measurements are based on this biological models. And we have 3 systems for indirect measurements, from biochemistry, hormone signaling, and similar approaches. Now, shortly to mention it. The first, this red one, is one of the most important photosynthetic activities of plant. So light and CO2 produce oxygen and sucrose. Sucrose goes through hydrodynamic to different organs. And so here we can measure, for instance, chlorophyll level, gas exchange. In our cases, we focus mostly on water exchanges, or ionic versus organic content here in the stem. So we can more or less measure directly a different product of these photosynthetic activities. It's not so, let's say, frequently used because you have to cover part of a leaf, but nevertheless, I mean, there are sensors and we can use the sensors. This blue one, this is one of the most, let's say, important and largely used in the phytosensing. Because generally in the phytosensing, of course, in our case, mean water. The water in soil is taken by roots, so water and mineral, water and ions. They go through stem to leaves and through transpiration is evaporated and then go back with sucrose as more organic solutions. Here, for instance, we can measure behavior of roots. Ionic contaminants, important points here is, for instance, heavy metal contamination. So ionic contaminants here, ratio between organic and ionic products. Transpirations. I mean, I will also later on a little bit describe the system in more detail with some examples. But like I said, it's one of the most important systems that we use in phytosensing. Green system is a biomass. Biomass, you know, plants grow, and typically they grow in the lower part, roots, and the upper parts it's a green biomass. Roots are typically difficult to sense because they are in soil, but generally we can sense root grow biomass and green grow biomass. This sensor is the most, the sensor is not simple, but interpretation of data is extremely simple. So, you know, you apply some approach and you just observe how fast it grows. Yeah. So, and finally, orange is electrical signaling. Plants, you know, living organisms, they live in the environment. They have to react in different stimulus. How do they do? They perceive this, I mean, for instance, mechanical stimuli, aeration, chemical stimuli, and so on. And for fast signaling system, they propagate it through an electrical way. No, and what we do, we call it a bioelectric response. We're just measuring a potential between different plant part of plants, so by different dipoles. So sometimes useful, sometimes less useful. This is one of the largest or oldest system ever used with plants because, I mean, like I said, it's very, very simple to use. Okay, there are four models, for models that can be directly accessed with different phytosensing systems. Now, three order that we can approach indirectly. The first one is chemistry. Chemistry is in plant, you know, it's pretty complex. Plants are extremely complex biochemical machinery. Typically, what we can measure everything related to ionic solutions. Ionic contaminations, Ross reactions, for example, hydrogen peroxide produce a lot of ionic reactions, so we can measure this reactive oxygen species. Once again, organic versus ionic, at least and this level we can access this chemistry of plants. Hormones, hormones are primary signaling system. Example is ABA hormone. ABA control stomata, opening and closing stomata, stomata changing transpiration, and transpiration change hydrodynamics. So if we are measuring here hydrodynamic and transpiration, this way we get access, or at least we can estimate dynamics of ABA hormone. In the same way, auxin, they control directional growth. And if we're measuring biomass in specific direction, so we are able to estimate at least some roughly dynamic of auxin as well. So in this way, like I said, it's indirect, so more exact or less exact, but we can, based on physiology, estimate hormonal structure and hormonal changes. And finally, circadian clocks. Circadian clocks define periodical activities as a 24th cycle, lunar cycle, and the year cycle. And whatever we're measuring here, we immediately discover the circadian clocks. I will show this in many diagrams. So whatever we're measuring, we immediately receive information about circadian clocks. So there are, like I said, 7 models, biological models, what we use. Now a little bit more how we can use these different biological models. First of all, we need electrodes. Typically, I mean, all electrodes, they are somehow combined in settings for specific applications. For instance, these are typical electrodes for laboratory on the table. So you have electrochemical sensor, root moisture sensor, transpiration sensors. Here there's another set of electrodes for vertical farms. So they are optimized for estimating biomass growth in this Young-Planck system. But generally, I mean, whatever you use, you need specific electrode for these purposes. This example, just this first set of electrodes is tomato plants. Tomato are one of typical model plants for many different purposes. And you install here 4 or 5 different sensors. It takes something like 5 minutes. And now you receive access to a large number of different sensorial data. They are generated once per second, or 1 per 10 seconds, or 1 per minute, it depends. And so you can start your analyzing. So in this way, I already say this, we call it sometimes this system as biochemical laboratory on table. It's one of the most important points here. This is ready for measurements for months. So you install different sensors, and as long as plant is alive, so you put a little bit water. You use capillary irrigation, so they don't need any maintenance, and the system is ready for measurement for a long time. So whatever you do, you can test your chemical technology or non-chemical technology, as the system is ready always for measurements. So, or as if you use, for instance, phenotyping with artificial intelligence system. So this system in a new table, you run some algorithms, and you immediately can receive and train your neural networks, for instance. So, to mention some, let's say, secondary details, we distinguish between 3 different technological, let's say, applications. If you use for 1-2 measurements, if you use for continuous measurement, or if you use for controlling vertical farms. Mostly what I recommend this case, as a middle case, if you use a continuously running mini PC as a small stick, it consumes, a little bit of energy, something like 6 watt of energy at all, and it's enough to run a phytosensor to collect data, to analyze data, store data, to provide, for instance, internet accessibility. It can generate HTML data, and you can see this data over the internet, for instance. There is regarding, let's say, in this case, you need this small mini PC or any mini PC. And the last point here to mention that in many cases, generally we distinguish between 3 different cases. So one case is outdoor application. If you, for instance, do like gardening or do in the field or greenhouse, that's all there are outdoor application. They require specific protection. So from at least from sunlight, and plants here are continuously stressed, especially by sunlight. There's then water stress. So it's sometimes difficult to measure. It's not only because of sensor noise, but because plants are in stress, and so it's difficult to measure. It's the first point here. Second case is indoor application. If you have some laboratory on the table or whatever, this very nice application, mostly you receive clear data. We like, of course, working in laboratory, but in many cases you can't do this because, for instance, you have some greenhouse or you have field and you have to measure out. You had to measure outdoor, but nevertheless, there it seems to look like a very similar application, but they are very different application. And last case, this vertical farming currently is very popular. Most of food, at least in Europe, is produced, at least not in summer period, but in winter period, is produced in this sort of systems. So there's here mostly young plants, so at least from very young stage to adult stage, and here different sensors, different model, and different approaches. So generally, if you use the sensor or you think to use the sensor, please think about these 3 different application cases, and so how to select electrodes, how to let's say measure plant physiology, stress physiology, non-stress physiology, just space for considerations. So now a little bit about different sensors. just to give you some flavor of applications. Like I said already, the most simple to interpret sensor is a biomass sensor. So biomass grows, and you can measure how fast it grows. And here, some amazing observation. For instance, if we measure it in lunar cycle, so we run the system vertical farm, and sometimes we see, well, in some period of months it's very fast. I mean, you really see a difference in growth in the biomass, in total biomass. But in some periods it's essentially less, so something like 20% less. And then if we done this sort of measurement, we really see that, for instance, in specific periods, so typically 2 weeks and 2 weeks. And the first 2 weeks you receive 20% more growth, and the second 2 weeks you receive 25% less growth, and you can nicely see this in the diagram. So this, let's say, nice application, nice data, and with the sensor you can really compare this biomass growth. Another point, not in a vertical farm, but for instance in this, with young plants, it's the same. You can measure a growth from something one day or 2 days after germination, and up to, let's say, 10-20 centimeters. So you can monitor developmental stage. You see, for instance, this plant grow faster, this less faster, and you can conclude why this grow faster, what approaches did you apply, chemistry, non-chemistry. You can make a sort of phenotyping and characterization. And like I said, interpretation of biomass sensor is extremely simple. It's either grow or not, and you can see a rate of growth rate. Same root biomass. Root biomass, for instance, here. You can compare, but like I said, biomass is difficult, as a root biomass, difficult to measure if you are in soil or in substrate, because they can receive fluids. And can store fluids in this media, and so it's difficult to measure. But nevertheless, there are some estimation to root biomass, so generally you can measure this. And one of nice observations, for instance, roots grow without light. If we switch off light, for instance, we see a much faster growth of root biomass. And so it's also another story, for instance, the behavior of roots, how quickly they can consume fluids. So you can make optimization of your irrigation based on this data, how much this tissue store these fluids. You can also work, if you have this diagrams, you can see how this or another light or irrigation or whatever impacts the biomass growth, and you can make a sort of conclusion, okay, how to improve this parameter or another parameter. Well, biomass, second one is a bioelectric signaling system. I already said this one of the most oldest, let's say, known approaches how to measure with plant, how to work with plants. Typically, measurement is very simple. You need a voltmeter, but specific voltmeter with high input impedance. And you connect with two different position in plant, and you observe first of all so-called action potential. Action potential is a very fast reaction of plant on different stimulus. Just by if you touch plant or there's a mechanical stimuli, plants inform the whole organism that something is happening, and so there's a very fast propagation. You can measure in different position of plants. So, and finally what you see a spike. So it's easy to interpret and easy to understand, and you can properly, for instance, touching and interaction with human, they can be only organized based on this action potential. More complex and less sometimes understandable is systemic potential, because it involves more biochemistry in plants. It's based on different unique content between cells. So it's mostly based on electrochemistry, and this very slow. So it's slow changing, and this the slow changing rate demonstrate different biochemical processes in plant based on stimulus. So stimulus applied, for instance, temperature is changing, and so the whole biochemistry slowly start to change, and you see this these changes in the systemic potential. So this systemic potential sometimes is useful, but there are two major problems with bioelectric system. This is a reason why basically, for instance, we almost don't use it at all. The first point is a plant is a large antenna, and whatever mobile phone signals or Wi-Fi router or whatever is in the vicinity of the plant, so everything start to interact, and plant of course receive all the signals and become very stressed. So all this electromagnetic spectra is reflected in this plant, and finally what you see is a absolutely chaotic signal. So first of all, is of course this electromagnetic stress produce stress on plant, but there's a reason why it's difficult to use. You have probably to apply here a Faraday cage, which is also not practicable in many cases, but nevertheless. This is the first reason. Second reason, plant physiology itself is very chaotic because there are large number of different stimulus. And for people, not for people, for plant, they react in all possible stimulant. You see a lot of noise here, and it's sometimes not easy to understand. Okay, what's happening? Because there are something like 10 different stimulus applied at the same time. Plant react always in the same way by changing this potential, and so finally you cannot really understand what's happening. So this is one of, for instance, reasons why we don't use it. But instead, we use electrochemistry, different electrochemical sensors, because they are much more stable, and they are connected to two major subsystems. So first of all, photosynthesis, and second one, hydrodynamics. The idea is here, this biological model what we use. This, in fact, propagation of fluids in stem. This is a stem here. And we apply two sensors in two positions, low position and upper position. And here you can think roots here pump fluids from soil with osmotic pressure. This go up to leaves. Here leaves, they evaporate fluids, evaporate water through stomata. And here, instead of water, we receive more organic fluids, and organic fluids go back. And so with these two sensors, for instance, with the lower sensor, we sense everything what is related to root, ionic contamination, root behavior, how much, for instance, roots can take fluids from soil, type of soils. Like I said, there are many things what we can measure only with this sensor alone. With upper sensor, the same story, but related to upper part of plants. First of all, of course, VPD, also vapor pressure deficit, temperature, humidity, light, photosynthesis, transpiration. All this is reflected in the sensor because they change hydrodynamic behavior of a plant. And like I said, it is one of the central transportation system. And finally, if you measuring something between both sensors, then you receive information about subflow, water status of plants, enrichment of tissue with water, and then similar issues. So, like I said, sometimes we use only electrochemistry, so this electrochemical sensor, and we don't use anything else, because here we get, I would say, more than 50-60% of information what we need to analyze dynamics of plant, also plant behavior. Some examples. This is measurements, electrochemical measurements over, I think, 20 days, so about 3 weeks, and you see very nicely changing of water status. So the slowly decreasing of water status, plant become over this time less and less water. It is visible here, for instance, decreasing, also increasing of impedance here. So you can analyze in this way long-term behavior. impedances react extremely well on irrigation stimuli, light stimuli, fertilization. I mean, everything what's related to, let's say, daily activities, and based on impedances, you can optimize illumination as a light, irrigation, fertilization, pH of fluids, and then similar story. So extremely useful system in practical application. Even we tested things like deficit irrigation based only on lower sensor, and you see, I mean, it's keep specific water content in tissue. So the point is you reduce water, but to keep some specific content in tissue so that plant do not die. And so we estimated we can save really something like 50% of water by keeping plant alive for a long time. So this only with impedances. So very useful sensor. This example, detection of stress and pathogens. In our case, this two picture what I already shown in ozone. Ozone is a pathogen, so air pollutant is extremely dangerous. And for instance, here, hydrodynamic system, this reaction of upper sensor installed here as a close to leaves. Reaction is something like 15 ppb, so per billion on ozone. If we started to compare with technological sensors, we didn't find the cheap sensor that are able to sense this level of ozone. So only a very expensive sensor. So plants are even in some cases better sensor than the technological ones. So, but these more or less examples how useful are different sensors, different let's say impedance-based sensors. And now I start to the next topic, last topic of the presentation, about different quantum phenomena. And here, important point is following. We had a lot of talk with, let's say, people from community, from farmers, from academia and companies, and commonly everybody say, nice, I mean, quantum is something, very small, very expensive. Usually we don't have access to this. But I found a really nice paper that summarize current trend in the quantum research. This for September 25. And I like this last sentence in the abstract. That, first of all, manipulating quantum effect in vivo. Now, this is exactly what we do a long time. We try to manipulate these quantum effects in vivo. And one important point, what we discover, typically small impact, small energy impact produce huge effects. And, it's a controversial in normal science. For large effect, you need large impact. And no way how with something small you can achieve something large. But in quantum, it's exactly the case. Typically, triggers that trigger some quantum processes are extremely low energy. And this low energy change dramatically physiology of a plant. And so this author of this article absolutely right. I mean, I fully agree that we start with agricultural applications. So, and I will at least show a few cases what we investigated so far, as not of all of them, but at least to demonstrate some let's say direction of this research. To give some overview, first of all, plant and quantum, it sounds like science fiction again, but the point is the whole biochemistry at the lowest level based on quantum effects. So, from this point of view, plant is a quantum being and a quantum organism, but we have specific cases. So at least I collected 4 cases. With 2 of them we can work quite, in quite good way, and for 2 of them probably not so directly. The first of one is for photosynthesis, well-known example. But what I observed, there is not photosynthesis, that we know from historical perspective. As a quantum photon change some reaction, and from this point of view, is a quantum process. Not originally, yes, but currently, if I make an overview about different processes taking place in this photosynthesis, I discovered a lot of things related to, for instance, quantum coherence. So it seems in the large area of leaves, all these processes are extremely organized, well synchronized, and point is properly here exactly we touch with this quantum coherence, and quantum coherence again mean entanglement. So properly here in the very large way we see different entanglement phenomena. But this is the first one. Second one, water transportation, and just two examples. These small channels in cell membranes, so-called aquaporine channels, and point of aquaporines, they are very, very small. This diameter of the channel is about 20 angstrom. It's, even a single molecule cannot pass through this channel. But here, water can flow through this channel. And the question is how, because water is, molecule is quite large. How it's happening here, and the point is only one of isomers. Currently, there is some discussion, of course, but it seems only one of isomers is passing through this channel because of this physical chemistry, magnetic properties of this isomers. This go more or less, through this channel by using these quantum properties. This is one of example here. Another. This again, this transportation system in plant. This phloem and xylem tissue. In xylem is water. So water is going up, and then after photosynthetic activities, all this sucrose and organic matter goes down and distribute to this different targets in plants. And one of the points here, this xylem tissue, they are very small again. There's something like the whole tissue less than 1 millimeter, and all the small capillaries, there is a lot of capillary phenomena. And what we, not only we, but in the community well known, both isomers heavily influenced by, or they as a change between isomers influence capillary properties, change thermodynamics, and so even small impact on plant, absolutely weak impact, can here in the hydrodynamic system create a huge change of physiology and productivity and behavior. So okay, there's a second. The third one is something again new, but not really new. This so-called cryptochrome. Cryptochrome is a molecule. Typically, it's also known in plant cryptochrome, animal cryptochrome. Typically, cryptochrome is responsible for sensing, especially, for instance, blue light sensing. And lately, it's known that it's in charge for magnetic perception, so it can sense magnetic fields. And here, people, as biologists, they wonder. Sensitivity of this cryptochrome is absolutely simple. They can even sense near zero magnetic fields. The number of publications near zero magnetic fields. And the reason how it's work, there is a Ross reaction, also reactive oxygen species between water and oxygen and hydrogen peroxide. And here, spins of electrons, through this extremely weak interaction, they change from signaling to triplet stage, and so they change properties of cryptochrome, and cryptochrome in turn involved in very different subsystems of plant, in circadian clock and signaling system. It means that any weak interaction with plant, like blue light or like magnetic fields, but we assume currently that not only this all weak interaction, like electrical field interaction, they change spin stage, also electrons here, and this heavily influence physiology of plants. At least we conducted some first preliminary experiments, and this I will show this. It sounds really promising. Okay, lastly, but we do not touch it a lot because this quantum biochemistry example is enzymatic catalysis in plants. Though it seems it is something, first of all, large area. It is not is in information now. So we know there's a spin chemistry, a spin catalyst. It's a huge area by itself. So at least I'm not risking even to make any overview. Just to mention, okay, there's a last field with this quantum biochemistry, but it's so large. But it seems the whole plant is based on quantum biochemistry. So. Okay, now question is what we can do, and because my point is we can work with this. We can measure this, we can influence this, and we see a visible changes, visible impact of the whole story. So plant is really a quantum biosensor, like in the title. So how to do this? There are two things. First of all, this water transportation system, we can directly work with this, measuring and impacting, and with cryptochrome. So this water subsystem. I already showed this slide many times, but once again, just to remind, water consists of two different isomers, and these isomers, difference only rotation of protons. So in one case, ortho isomers they rotate in the same way, and in para they rotate in opposite way. Sounds like once again it's the same water, so water molecules. Point is, on the chemical side, they have very different reactivity. So they have different chemical reactions, so speed of chemical reaction, how fast they can do products from, let's say, one product to another product. So how fast they can do. This is a chemical reactivity. And this is exactly what we sense, at least in the mine technology and all this, what we use impedance spectroscopy. Exactly, we use different chemical reactivities here. But it's not only different spin isomers, they have different surface tension, viscosity, they completely differently react with a magnetic field. For instance, ortho isomer only react. For instance, thermodynamic behavior is different. This is on chemical side. But we have also a quantum side. And so now this ortho and para isomer is absolutely nice sensor between chemistry and quantum side. So whatever dynamics is appearing here on quantum side, we can measure on chemical side. And here point is, this is not expensive technology. Here in the chemical side, we can measure with impedance spectroscopy, with optical spectroscopy. We can measure even with some simple, like measuring viscosity. You can do this with capillary tubes. So it's a very, let's say, not cheap, but you have to measure this. You need a laboratory, but it's not something like for one sensor you need €1,000,000, yeah, for MRI tomograph, for instance. So it can be measured by normal means what we use in normal day-to-day practice. So a few examples. The first one, like I said, we started with a hydrodynamic system, and I think that everybody conducted this sort of experiments. And so we demonstrate this experiment from our side, but probably everybody already discovered the same phenomena. So point is here. If you apply weak excitations like electric field excitation, magnetic field excitation, if you apply something absolutely almost not measurable, you discover, at least with plant organism, large changes. And we try to apply, of course, to explore this behavior. We use these capillary tubes here. These very thin lines, the small capillary tubes, about 0.3 capillars and 0.5 capillars. This is very simple to xylem tissues. And what we apply, if we, for instance, use hydrodynamic cavitation or we do magnetic field treatment, we see how it changes capillary forces. Here, for instance, if we apply hydrodynamic cavitation, so we see that in one tube, capillary force is much larger, about 8 millimeter larger, so the growth of this capillary tube. So it means here in the xylem tissue receive more water and water propagates faster through the stem. And of course, this creates as a second order of reactions and plant physiology and, for instance, for water consumption, for biochemistry, because you applied something absolutely weak and absolutely simple. Now another point, and this sometimes, I mean, we cannot explain this, but reaction, what I will later. But the reason of this reaction is change of thermal capacity. You know, thermal capacity is something what is not easy to change, because it's a level of properties, very basic properties of materials, and thermal capacity is not, let's say, so easy to change. But if you, what is the same way, what we done hydrodynamic cavitation, and then measuring thermal capacity of this water. And then we see the thermal capacity change dramatically, but for a short time. It's about 1 hour, 40 minutes, or hour 20, something like this, not for a long time. And so in this time, we observe a lot of crazy phenomena with water, with plant, if we apply all these small changes. And finally, what we saw as application of these examples, capillary effect, the thermal capacity, water status, water enrichment, and explanation, at least our current working hypothesis, this low energy spin conversion at interfaces. The point is, this spin conversion, just what is spin conversion, is spin change rotation from, let's say, ortho to para or vice versa. Energy required for this conversion is extremely low, something like estimation 10 of minus 26 electron volt. It's absolutely, I mean, simply, I don't know the level that usually even temperature produce something like 100 more intensive effects. So, but point is that each interaction, molecular interaction, spin change our state. And so our assumption is, and with colleagues, we think it's happening at interfaces. Interfaces, a water-air interface or water-metal interface, or here a lot of interfaces, water membrane, water biological object. In any cases, if, for instance, you have a sort of ice-like structures, and they fix water and preventing interaction between isomers, for instance, or between water molecules. So in this way, we can observe this phenomena, and they in turn change all these chemical and physical properties. At least this we published, finally, paper we received 2 days ago that is finally accepted in experimental thermal and fluid science. So we will, share this research in the community. I mean, absolutely nice results here. What we also discover, magnetic fields. This is fluids, and we took a classical reaction, this hydrogen peroxide with water, it produces ions. And if we apply a weak magnetic field, 500 microtesla. You know, Earth magnetic field is about 70 microtesla, and more or less in the level of Earth magnetic fields. But if we apply this control example, and this is this experimental fluid, fluids start to behave absolutely different. So this reaction, it's not, so just you know, if you have fluids, or you have a plant with fluids, or you have other biological beings, and you apply weak magnetic fields, even this Ross reaction, they start to behave completely different, in such different ways that they start oscillations. So this exactly the same way, this pure water, we put magnetic field treatment and pure water, and you see that some hours, probably about 1 day after excitation. So 1 day after you applied something extremely weak and your fluid samples or your organic samples, and what we see, for instance, here, this region, you see oscillations of electrochemistry. And this ongoing oscillation. So if we publish this, but we come to some biologists, chemistry people, and ask, okay, what do you think about how it's possible? Because it's pure water. I mean, pure water cannot oscillate in normal conditions. Yeah, it's just some relaxing behavior. You see some changes around equilibrium point, but not that reaction oscillate over 1 day or even longer. And currently, there is no good explanation why oscillation happening. But especially important is we observe not only oscillation of electrochemistry, but we observe thermal oscillations. And this returns us to original point, what I said, ortho and para isomers, they change not only electrochemical reactivity, but they change thermal capacity. So they influence all these endothermic, exothermic reactions, and they in turn change electrochemistry. And so it seems that sort of interdependency, changing of thermal conditions for a reaction, that change equilibrium condition for fluids, they change in turn ions, they turn again thermal conditions, and so we receive a sort of interdependency between thermal behavior and electrochemical behavior. And it is our current hypothesis, and so it's a periodical reaction taking a couple of hours. And now think about plant organism. So you apply weak magnetic field in, of course, specific conditions, and then plants start to oscillate, or is going to die, or going to feel well. It is depend, of course, on the physiology, but you applied, like I said, for a very short time period. It's about 1 hour here, and something like 500 microtesla. And you see a huge change in the dynamic of living being. So, okay, this was one example. I think exploration of water in organic tissues, at least with phytosensing, you can measure this, you have direct access. There's a lot of things what you can do. But the question is, because quantum, or at least spin level phenomena, people always skeptic, think, okay, can you measure this directly? And answer is, of course, sure. There's a well-known effect called nuclear magnetic resonance. It's exactly spin-based measurements. And everybody say, well, MRI is something, 1 million tomograph and large device, but no. For measuring MRI, you need only 2 coils. One excitation coil and one sensing coil. Now, there is something absolutely, simple. There is a lot in the community. There is even, a large community doing a self-community how to create a nuclear magnetic resonance sensor. It's something like this, has a couple of coils. In fact, we following this developmental line. You can, for instance, here in the plant science, you can use permanent magnets. You can use, for instance, just a moment. You can use this permanent or electric magnets here. You put plants, for instance, inside of the first coil. So it's, not the technology. There are different technologies. As a whole development called portable nuclear magnetic resonance, and there is a specific version related to Earth magnetic fields. This overview over state of the art is not our research. But for instance, here with this device, like I said, it's only a couple of coils. People are able to measure hydrogen resonances. This for hydrogen, and it seems they claim, okay, that's a signal to noise is not good, but it can be improved. There are different directions. Here you can measure chemical shifts, and chemical shift is the most important because here you can measure all this chemistry. You can identify exactly substances in chemical reaction that have hydrogen. So you can identify, not only some qualitatively measure, but you can identify different molecular substances, and you can characterize chemical reactions. And this one of the modern, let's say, instrument is MRI and NMR approaches for plants. For instance, what people demonstrate is circadian cycles, and I assume there's not only water, because here people claim, okay, there's a circadian cycle of water, but MRI, it is important, is measuring only one of isomer. So it's only measuring orthoisomers, and here we see a variation of 2 parameters, water content and of isomers. And now the idea is basically what at least we think, if you fix water content or measuring water content in electrochemical way, you can easily measure here orthoisomer as one of the isomer in the water or in plant tissue or in whatever. Another story what also well known, for instance, which tissue contain water in plant structure, and here you can see it's only a few millimeters water containing layer in plants, and then everything the remaining it's the different functionality of tissue, but water content is only this, a small outer perimeter of plant. They only contain water. Well, we work on this sensor quite a long time. Like I said, it's not essentially difficult, but problem is there's a timing, excitation timing, where spins start to react in different way, and you can measure this reaction by changing of magnetic fields. So, for instance, orientation of coils in this way, or for instance, Helmholtz coils, and we discover here one interesting phenomenon that I would like to share now. And this is related to so-called collective reaction of plants. So, what are collective reactions? This publication, 25, is really recent publication. People claim, and by the way, probably you also know many people from, for instance, Giuseppe or Andrew Adamaschi. I mean, they're well-known people. And what they measured, trees, bioelectric reaction of trees, and it seems that tree can react on solar eclipse, but now the point, not only after eclipse, it's happening on sun, and then electromagnetic emission or particles, they enter to the Earth magnetosphere, and everybody can feel it. No, in advance, before solar eclipse happens, plants starting bioelectric reactions. And this alone, this is crazy enough, And the journal is good. It's a Royal Society, and like I said, people are well known. So plants capable to sense cosmic events in advance before they happen. So they will happen. Probably there's a sort of emission, and they or whatever. I mean, but basically we don't know how it's happening, but in advance. But another story is even more crazy. What people discovered, reaction is collective reaction. All plants, they react in collective way. It seems there is a sort of synchronization between plants. So every plant, or probably only some plants, sense this. They say, well, that's most all. So I really recommend, please read paper. It's an extremely nice paper. So probably only some three can sense the solar eclipse, and then they propagate this reaction through communication. And now we start this collective reaction of plants, and currently nobody can really explain. I mean, why plant can communicate and how it's happening at all. Yeah, this communication between plants. And finally, there is, of course, very controversial discussion, but I mean, this measurement, you cannot say it is a fake measurement or something like this. So they really measure it. And there is a long discussion how plants can communicate really in collective way. Because, this once again, we encounter the same story, there's something weak. And for communication, you need a lot of energy here. But plants cannot produce energy, so much energy. And so the question is how they can communicate by something weak, but though that weak enough, so at least this weak is strong enough to communicate over some meters of distance, or no, this is currently unknown. But we of course assume that something like interactions between spins and this quantum level, and we demonstrate that in fact there is sort of macroscopic entanglement happening heavily in fluids. I already demonstrated multiple times this slide. This published in 25. And what we saw that electrochemical reactions here, they can be synchronized. They have so-called synchronization events. And it means all these separate water containers, also electrochemistry in water containers, that start to behave in extremely synchronous way. The question is a sort of entanglement, but question is in which way it can happen. And our hypothesis is it's probably light. We call it photon-assisted entanglement, or even we also think currently that's probably also electromagnetic emission. So electromagnetic emission can create a state for a short-term synchronization or quantum coheration between different molecular issues. But okay, point is, it's quite easy to measure. So you're measuring electrochemical reactions, and you see here a spontaneous change of electrochemical reactivity, and this is exactly due to spin conversion. So it seems that spin conversion, different molecular oscillator, they start exactly the same way to produce more para isomers. More para isomers, for instance, produce more intensive reactions, and we see here this more intensive reaction. So we calculate Pearson correlation coefficient, and we see, okay, very high values. Absolutely technical work, but what we also performed, we performed the same measurements with plants. So these are plants, tomato. You know, typically what we do, we also measure electrochemical reactions. And what we basically need, we only need to make a correlation analysis between different measurements, between different plants. And in normal condition, we do not see any, let's say, huge correlations. So for these correlation coefficients, you see it's like behaves normally between, let's say, up to 0.4 something. I mean, there is no correlations. But at some conditions, we observe extremely highly correlated electrochemical dynamics. So, and here, for instance, correlation, it's become 0.85, 0.9, 0.92. It means here something is happening. And all this electrochemistry of separated plants, and you see plants are on the table, as in laboratory, distance something like 20-30 centimeters, as large enough. But electrochemical reaction, they start to synchronize, and we assume phenomenon exactly the same. Properly, it's a synchronization of chemical reactions, but happening in plants. By the way, this collective reaction of plants is well known. We are not the first one who discovered this. I think the first one was Maslabrod in Institute of Genetics. He published this in 1991. And he called it a sort of plant transformator. So without, let's say, evident magnetic fields or any reactions, plant, as the bioelectricity of plant become very synchronized, and it keep for some time. So effect is known, but currently no explanation, and we also don't know why it's happening, what are conditions for happening this. And then finally, what we heard, there is this phenomena with cryptochrome. Cryptochrome, you know, they use spins in electrons. Just to explain, this paper that I refer currently. Here is this Ross reactions between hydrogen peroxide, and this weak interaction, what it change spins, electron spins from triplet, from singlet to triplet state, as a sort of spin conversion. But energy for this change is low, and currently blue light and magnetic field. But we assume that you can change it based on a magnetic field, or you can change by adding energy, optical energy, or for instance any other form of energy you put to the system, and this increase this levels, energy levels. But point is, all the spins they can also enter in the entangled state. And the idea is that properly plant cryptochrome could be responsible for this interplant interactions. So just by using, there is a well-known idea with spin chains, and entanglement in spin chain creates some crazy behavior on this spin dynamics. And so we assume basically we encounter the same phenomena with plant cryptochrome. But most important is here that if plant cryptochrome is involved, we should observe a physiological reaction. So it will answer, let's say, our question here, if this just a synchronization of spins in para or ortho isomers, or the synchronization of electron spins. And finally, what we have done, There's one of really nice experiment, this nice work, what we conducted with colleagues, but I mean background is different. But here we applied magnetic stimuli, one of our test approaches, and we measured physiology of the 6 plants. They are more or less spread in the range of 1 cubic meter on the table. This very clean laboratory, I mean, from encapsulated from all these electromagnetic fields, low level of electromagnetic emissions. But here we still have phytolite. So from phytolite we have magnetic field and electromagnetic emission, of course, and optical emission. And the question is, if this really are cryptochrome and all these environmental stimuli can generate entanglement between different spins, electron spins, that probably we will see a collective physiological reaction. And what finally we done? There is local excitation of a single plant. We tested either with pepper or with tomato. We put here the very, very small stimuli, magnetic stimuli is about, let's say 500 microtesla, 800 microtesla, 200 microtesla, in this range. It seems, if we have, at least we think this phenomenon has a resonant properties. For instance, we don't know exact frequency, so we started from microhertz to from millihertz to megahertz. It seems that in some cases doesn't produce any response, but if we sweep, for instance, from micro to mega, we observe a collective reaction of plant on the table. And this you see this clearly physiological reaction. Plants switch from one physiological stage to another physiological stage. You see it here very clearly, and this is quite well repeatable. For instance, we performed this a couple of times. If we use exactly the same condition, the repeatability is quite high. So at least this one of, let's say, first preliminary experiments. We can explore this. We will definitely explore this in more details. But one of example how we can use and we can see this quantum phenomena. We can work with them. And I think there is a great potential in agriculture in all cases, because here, once again, you apply extremely weak stimuli, magnetic stimuli, and you can change hydrodynamic state of plant, and not only local plant, but all plant in the environment. Nice. Okay, I think we go now to conclusion. My first point, phytosensing is great. It not only because we do this or we deal with this, because this really great technology. In indoor case, for instance, you have a complete biochemical laboratory and you table. And there is not only, water research. People sometimes, work only with water as a sensor. Here you have a complex organism, and you can measure different reaction in this organism, physiology, electrophysiology, transpiration, photosynthetic activities, and you have a huge amount of data. And now if, for instance, you do this AI phenotyping, also you use artificial intelligence to understand plants, just in your table that generate you probably megabyte of data per second or per minute, how quickly you sample data. It's produce a huge amount of data for training purposes. Biosensor, plant as biosensors. Plants are extremely sensitive, even to classical or non-classical stimuli or events. If somebody works with plant, how sometimes they demonstrate strange behavior, strange effects. So plants are extremely sensitive, and you have this sensitive object on your table. You can work with this. Install 4 sensors. 5 minutes, and then 4 months you can experiment with this. This is great. But in outdoor conditions, there's B2B cases, improving productivity. Like again, improving of productivity, you can achieve something like more than 200 times. And this is huge, at least in agriculture in Europe, with all this climatic changes, water deficit, and similar stories. This improving productivity is a huge thing. Stress and pests, the same. You're measuring stress immediately, and you can prevent pests. You see happening something in plants, and probably plant will die in 2 weeks, and you see this now. You can now prevent so that will not happen. Okay, water deficit. So from this point of view, I think phytosensing is really great. For somebody who use already phytosensors, please think about biological models. Of course, you receive a huge amount of very nice graphics, but you need to understand data, to interpret data in terms of their physiology. What, for instance, you apply some informational effects, or you applied magnetic stimuli, or electrical field, or whatever. At least we tested in laboratory, probably, I don't know, many of them. But now we know it's work, but it's not enough. We need to characterize what is changing. That changing hydrodynamics. Plant receive more water. It's more biomass grow. So if biomass grow, for how much larger or how quickly? I mean, all these parameters you need to characterize. Use biological models. It's extremely important. At least what community, as not only we as a community, provide 4 models for direct measurements. Please make somehow learn about this. It's not so complex, and use them. And 3 for this indirect measurements, but it's little bit more advanced topic. But still, I mean, if you can, for instance, work with circadian clocks. And from circadian clocks to signaling system, plant hormones, cryptochrome, and other story. I mean, there's a great advance in the whole community. And my last point, of course, quantum effects in plants. They happen. We see them. There's all these stories where if for instance some huge effect on weak impact, and in previous time we receive many stories about it's something not really scientific, because something we cannot generate something huge. It's impossible. I mean, it's something like 10 years ago. There was, if you try to publish this, there was a typical review. Guys, you made some error, so mistake in the measurements. You have artifacts. Please think again. Go. But now we clearly see as a weak impact can generate huge response. And this is one of this point of course, quantum phenomenology, and we currently think it could have a huge effect in agriculture, especially by weak magnetic field or by weak whatever story. You change behavior of number of plants, so you don't need too many sensors or too many these devices in the field. So I think that is all from my point of view. Thank you for your attention in Zoom in the seminar, in YouTube, who will watch this video after the seminar. Please contact us if any questions. If you need more information, you can find on our homepage. We will have other upcoming seminars in 1 week and 2 weeks, so every Saturday at 4 in Central European time. Extremely nice topic. Please attend the seminars as well. So now, if there are any questions, I will try to answer them. Okay, thank you.
Thank you.
Maria, can you hear me at least? Vlad? Okay, you can hear. Okay, fine.
Yes. It was okay. It was perfect.
But okay, if no more questions now, then I would finish this seminar. If any questions will appear later on. Please write us comments, for instance, in YouTube, or write a direct email to us. So,
hello, sir.
Yeah, hi.
Sir, my name is Zaheer. I'm from Taiwan.
Yeah, hi. Nice to hear you. Yeah. You managed to attend, yeah?
Yes, sir. Sir, it's very informative today's seminar. So I learned many things today. So I request you, can you make next section how to use this device? I mean, if I want to measure biomass growth or effect of light or like this, so which electrode or which setting should I use?
Yes. I know. First of all, many thanks for attending. You have currently probably deeply night, yeah, in Taiwan.
Yes.
Thank you. By the way, you are invited to make a seminar because what you do is absolutely, I mean, absolutely interesting. Your latest experiments. To answer your question, yes, we already prepared for at least some basic setup. Um,
Yes.
problem is languages. We have many different languages, or we prepare now in one language, and for probably
Okay.
only the first step for using like impedance spectroscopy or biosignals, how to put electrodes, everything. So it's still in progress for biomass. For biomass sensor, for clip sensors, because you know sensor what you use as needle sensors. So if you use clip sensors with strong needles, probably also they will also useful for you. I don't know, but a little bit different. There are different materials for needles and plant tissue. They react sometimes more allergically or less allergically. So this depends currently on species because it's not silver. So silver, is very weak. So you use stainless steel or brass. So and this create a different reactions. But yes, I mean we definitely will do this. Probably in this summer we definitely will prepare on biomass and on using clip sensors. Yeah.
Okay. Thank you, sir. Thank you so much. Okay.
Okay, but if no more questions, okay, then still, I mean, please feel free to write in chat or in YouTube or direct email, and then I invite you to the next seminar that will take place next week, next Saturday. It's exactly the same time. So it's absolutely nice topic. I really, I mean, look forward to see this presentation. Okay. Okay, then see you next week.